Wow, welcome. We made it to Unit 3. Unit 3 begins with a very interesting textbook chapter, chapter 13.1. And in this chapter of the textbook, it actually goes into all of the scientific experiments that went down in order to discover the structure of DNA and to determine that DNA was actually the basis for our genetic information. So, you probably already know now that DNA looks like this double helix formation. However, it took scientists a long time to actually figure out that this is what DNA looks like. So this section is kind of just going over the experiments that happened for us to decide that this is what DNA looked like. Um, and at the beginning of the 20th century, so this is actually pretty recent knowledge in terms of the grand scheme of the world. As early as the 20th century, we still didn't know what caused inheritance in biology. Some people thought it was proteins, and some people thought it might be nucleic acids, but we still weren't really sure. Um, so we did discover that there were genes located on chromosomes, and we knew that chromosomes were made up of DNA and protein, but we didn't really know which part was really responsible for the inheritance of genetic material. And we basically studied bacteria and viruses, and that's kind of how we started to figure out, oh, this is where DNA comes from, and this is how DNA works. So the first really transformational discovery that we saw about DNA was done by a research experiment by Frederick Griffith in 1928. And basically what he did was he took two strains of pneumonia bacteria, one pathogenic and one harmless. So pathogenic means it carried the disease and the other one did not. And when he basically took the pathogenic material or pathogenic bacteria, he heated it up to a very high heat, killed the pathogenic strain, and then he mixed that pathogenic strain with living cells of the harmless strain. And what he noticed is that some of the originally harmless living cells became pathogenic, meaning they acquired the genes to pass on the pneumonia disease. And so we still observe this phenomenon today, and it's called transformation. And this is a change in the genotype and phenotype of a particular cell, like their gene expression, due to the assimilation of foreign DNA. So basically, we discovered that bacteria can be transformed in which they accept and can get DNA from another bacteria and share DNA with each other, thereby changing the way that they express their genotype or phenotype. So in this case, the non-diseased bacteria were able to become transformed with the diseased bacteria's DNA. So this is just kind of an example of how they might have done such a thing. So they have our rats and our pneumonia. We've got our living cells that are pathogenic as a control and living cells that are non-pathogenic as a control. The pathogenic cells inserted into the mouse dies. The living cells that are non-pathogenic, the mouse stays healthy. When they killed the pathogenic cells, the mouse was healthy. But when they mixed the heat-killed cells and the living cells, the mouse died. So that's kind of an example of how that experiment worked. So later on, they weren't really sure what was causing this transformation, but later on, scientists discovered that this bacterial transforming substance was the DNA. But scientists were still not convinced that DNA was the main method of transferring genetic information because they didn't really still know a lot about DNA. A lot of it didn't make sense to them, and they were like, no, proteins just make more sense. So we got some more evidence for DNA being the genetic material by studying viruses. And the viruses that infect bacteria are known as bacteriophages, or phages for short. And they are used for a lot of molecular genetics research because essentially all that these type of viruses are is a DNA or RNA enclosed by a protein coat. So they're very simple organisms, and in order to thrive and live, viruses have to infect a host cell and take over that cell's metabolic processes in order to allow themselves to reproduce. So we can study DNA in viruses because in order to kind of take over and infect something, they have to infect another cell with their own DNA. So it's very easy to trace. So this next experiment that we saw was conducted by Hershey and Chase, and the main purpose of result of this experiment was that it showed that DNA was the genetic material, and they particularly studied a phage known as T2. So they designed this experiment in which they were trying to figure out whether protein or DNA is responsible for reprogramming the host cell. And so what they did was they had radioactive phages, and they tagged the two phages radioactively. So phage number 1, they tagged sulfur, and in phage number 2, they tagged phosphorus. Why did they do this? Well, because as you remember, many amino acids have sulfur in them, but DNA, nucleic acids, do not have sulfur. So by tagging the sulfur, that was how they would keep track of the protein. And by tagging the phosphorus, they would be able to keep track of the DNA, because remember, the sides of DNA we know to be sugar and phosphate groups. So in the radioactive protein phage, they infected non-radioactive E. coli cells with this T2 phage that had the protein marked. They put it in a blender, essentially, mix it all up, break it apart, and the phages are able to release their DNA into the cell, or the protein rather. And then basically what they do is they look at the end result and observe that sample. They do the same thing with the DNA. So they have the labeled radioactive DNA phages. They let them infect the cells. They agitate them so that the DNA can be released into the cell. And then they take a sample and they observe the end results. And what they found was that for the phage DNA, the DNA entered the host cell, whereas the protein, the phage protein, did not enter the cell. It was found in the extracted liquid, so it was not going into the cell. Whereas the radioactive DNA was found in the cell. So essentially, the Hershey and Chase experiment showed us that the phage DNA entered the host cells, not the protein. The protein did not enter the host cells. And so this helped prove that DNA was the hereditary material. There was some additional experiments that went on at this time as well, because at the time we did know that DNA was a polymer, and these nucleotides that made up the polymer consisted of nitrogenous bases, sugars, and a phosphate group. So there was a man in the 50s named Erwin Chargaff, and he had observed the DNA of a lot of different species, and in doing so, he realized that different species had different DNA. So example, a snail DNA looked different than a fish DNA. And that was a good sign that DNA could be the genetic material, because one reason people were skeptical is because they didn't think DNA had much of a varied structure, but they noticed that there was so much genetic variation in the world, they were confused about how DNA could be that, how DNA could do that. So Chargaff helped with that, and this made that claim a little bit more credible. So this is a picture of what we know DNA to look like. Right here you see the sugar phosphate group sides, and then the nucleotides, nitrogenous bases coming together. And here's another picture as well. We also realized that Chargaff realized that the percentages of A and T bases were equal, and the percentages of G and C bases were equal. And so even though the order of these bases was different between each species, the percentage of each amount was equal. And we will understand why that is in a little bit. But these became known as Chargaff's rules. So these two rules right here, the fact that different species have different combinations of DNA, and then that no matter what the species, the percentage of adenine and thymine bases are equal and the percentages of guanine and cytosine are equal. These are Chargaff's rules. And they helped us understand more about DNA as well. So the people you probably have heard of are Watson and Crick, as well as Maurice Wilkins and Rosalind Franklin. And these are the folks that we really owe to helping us understand what the structure of DNA actually looks like. Remember, structure always influences function, so it's important for us to understand the structure. So Wilkins and Franklin used a technique called X-ray crystallography to study the structure of different molecules, in particular DNA. And in doing this, Rosalind Franklin was able to produce a picture of DNA using this technique. So this is a picture of the X-ray diffraction, and essentially what this is showing is this is not what DNA looks like, but these spots are produced and smudges are produced by diffracted X-rays. And so those individuals are very familiar with X-ray diffraction, understand the patterns that this results in the structure, and that is what really confirmed that the DNA was a helix. And then they were able to see that based on the spacing and the pattern in the photo, that not only was DNA a helix, but it was also a double helix made up of 2 strands. So this is what we believe DNA to look like. Actually, it's kind of what we've deduced it to look like for sure. And then we know that it has a 5' end and a 3' end, which is known as anti-parallel structure. It has a sugar phosphate backbone. So you can see here's our sugars, here's our phosphates, phosphate group, back to functional groups. And this is really beneficial because if you notice, these phosphate groups have a negative charge on them. So remember, anything that's charged or polar has a structure that allows these molecules to be hydrophilic, and anything that is nonpolar would be hydrophobic. So the phosphate sugar groups are polar and hydrophilic, and then these nitrogenous bases are in the middle because they are protected as hydrophobic areas of the cell. So the DNA can be an aqueous solution because these hydrophilic ends protect the hydrophobic bases. And that's kind of what we've determined the structure of DNA to look like. So Watson and Crick, they built these structural models in 3D form to really match up that with Franklin's X-ray crystallography. And like I said, this molecule, they found to be anti-parallel, so meaning that the sugar phosphate backbones run in opposite directions. And that is important because, as you can see, the nitrogenous bases are attracted to each other because of hydrogen bonding between these different bases. And so they discovered also in doing this that the bases did not pair similar to similar. And the reason for that is because if you pair the bases similar to similar, they don't end up in a uniform width. Instead, they need to pair a purine with a pyrimidine, and that's what's going to give us a uniform width. So if we go back to this image, our purines are those that have 2 rings, so adenine and guanine. And then our pyrimidines only have 1 ring, thymine and cytosine. So if we tried to pair 2 thymines together, it would be 2 1 rings, but if we paired 2 adenines together, it would be 2 2 rings, and so it wouldn't be uniform across the whole structure. So if you had 2 purines together, it would be too wide. 2 pyrimidines would be too narrow, but they discovered that a purine and a pyrimidine was really consistent with the X-ray data. And so in doing this, they determined that adenine always paired with thymine and guanine always paired with cytosine. So adenine and guanine are our pure rings, the 2-ring structure, and thymine and cytosine are our 1-ring pyrimidine structures. And this gives us a uniform example. This, therefore, explains Chargaff's rules, because if you remember, he found that certain nitrogenous bases were found in equal ratios. And that makes sense, because however many adenines you have, because they pair with thymine, you have to have the same amount. And then same for guanine and cytosine. So the Watson and Crick model of the structure helped us determine the function. The sequence of the DNA, still with Chargaff, it could be in any order.